first technique to modify the DNA bases in vitro, it is cassette mutagenesis. Now, mutagenesis, it is very basic tool in the field of molecular biology. जैसा कि मैंने पहले बताया कि इसकी मदद से हम जो मुख्तलिफ जीन्स हैं उनके फंक्शंस को स्टडी कर सकते हैं. We can compare that this particular gene, it produced this particular protein, and after inducing the mutations in that particular gene, we can compare the function of a protein in relation to wild type phenotype or the bacterial strain spores. So it is very important to know the functions of different gene. And classically, these mutations, as I mentioned earlier, they were induced by using different type of mutagenic agents or physical factors, and commonly they may be called as the mutagens. And those mutagens, although they were used, but classically they were very important because it led to the development of the field of molecular biology. Although the significance of those mutagens were very important, but they were having certain disadvantages. For example, at the beginning of the process of mutagenesis, under the laboratory conditions, what was the problem? That whole organisms like the bacterial cells or some higher organism like the fruit flies, in order to induce the mutations, they have to be exposed to the radiation. So whole cell or whole organisms has to be exposed to the mutagenic agent. So in this way, the other non-target genes, they may also be mutated, and we also have no idea that whether the target gene it is mutated, and if it is mutated, at which frequency. So different screening strategies were required for this purpose. So this is one of the disadvantage. Another disadvantage was that if after mutation the desired phenotype, if it is available, still we don't have any idea that whether the mutation it is within the desired gene or target DNA segment. And third disadvantage was that before the availability of modern DNA sequencing techniques, we don't have an idea that whether the mutation it is within a gene, and if it is within a gene, which portion it is mutated, and whether this mutation it is due to base substitution, addition of DNA, or deletions. So we don't have informations by using classical mutagens. Then with the passage of time, the availability of modern molecular techniques or sequencing techniques, now we can induce mutations under the laboratory, and in such a way that a specific base or segment of the DNA it can be targeted or altered. No, because we can target a particular base within the DNA. So because of this reason, this laboratory process it is called as site-directed mutagenesis, and one of the type is cassette mutagenesis. Now what we can do in cassette mutagenesis process, here the target gene, if it is present within a recombinant vector, it can be mutated. For example, for this purpose, if target gene it is present within a recombinant vector, we must have two target sites that may be flanking our target DNA, so that by using restriction enzyme, the DNA fragment of wild type it can be removed, and then it can be replaced with synthetic DNA fragment. So in this way, we may have DNA alteration efficiency maybe around 100%. But the basic prerequisite is that target sites for restriction endonuclease, it must be available so that we can remove the wild type fragment of the DNA and replace it with our own synthetic fragment with our own choice of sequence. How it can be carried out? Here it is the basic outline that we first have a recombinant DNA plasmid that contain our target gene here, that is shown here maybe as a pink, and then by treating with restriction endonucleases it can be removed from the vector and can be replaced with synthetic DNA fragment that contain our sequence of choice, and then it can be ligated with the help of DNA ligase. So in this way you can say that the mutation efficiency it may be 100%.